Scientific advances in whole genome sequencing proceeded through three major technological revolutions. First-generation sequencing, next-generation sequencing, NGS or high-throughput sequencing. Third-generation sequencing or single-molecule sequencing. These three sequencing methods, however, differ from each other in terms of cost, runtime, and detection methods. The first-generation sequencing came on board in the 1970s. In the first-generation sequencing, there are two major methods, the Sanger sequencing and the Maxim-Gilbert sequencing methods. However, among these two, the Sanger sequencing method is more recognized. A major limitation of the Sanger sequencing method is that it is only capable of reading a sequence of about 500 to 800 bases long. The NGS technologies, on the other hand, provide massively parallel analysis, extremely high throughput from multiple samples at much reduced time and cost. In the third-generation sequencing, the sequencing of single DNA molecules occurs without the need to halt between read steps, whether enzymatic or otherwise. The third-generation sequencing emerged to reduce the price of sequencing and to simplify the preparatory procedures and sequencing methods. There are three categories of the third-generation sequencing. First, sequencing by synthesis method, whereby base detection occurs in real time. In the second category, nanopore technologies, DNA thread through a nanopore and are detected as they pass through it. And lastly, the third category, direct imaging of DNA molecules, a method that uses advanced microscopy.